Hello, everyone. Welcome to today's webinar titled A Systematic Approach to Define Operating Space and MRT for Continuous Manufacturing Tablets, Opportunities and Challenges, hosted by BioPharma Webinars and presented today by Dr. Ahmed Zidan, who is Senior Staff Fellow Pharmacologist, Center for Drug Evaluation and Research at FDA. So firstly, hello, Dr. Ahmed. Welcome. Thank you for joining me today. It's a pleasure to have you here.
Hello,
Okay.
Marcus.
Thank you. Hello. And following Dr. Ahmed, we're also joined by Owen Rehrauer, who is the president at Sentronic US Corp. So, hello, Owen. Welcome. Thank you for joining us today. It's a pleasure to have you here as well.
Thank you so much, Marcus. Thank you for having me.
It's a pleasure. And then last but not least, we're also joined by Rodolfo Romanach, who is PhD Center for Structured Organic Particulate Systems at the University of Puerto Rico. So Rodolfo, hello, welcome, and thank you for joining me as well.
Hello. Thank you very much. Thank you very much.
Thank you. So my name is Marcus. I'll be today's moderator. And before we begin, I'd just like to inform our viewers that we will be having a live question and answer session after all the presentations before at the end of today's webinar. So if you have any questions, then please feel free to send in as many as you like. Via the questions tab, you can find this directly below your webinar screen. And we look forward to going through your questions together with our presenters later. Okay, and please also include in your questions if they are meant for Dr. Ahmed Zidan or for Owen or for Rodolfo. Okay, that helps me direct the questions. Okay, now please allow me to introduce our first presenter. So, Dr. Ahmed Zidan is a senior pharmacologist staff in the CDER/OPQ, and Ahmed has a PhD in pharmaceutical sciences and leads the Advanced Manufacturing Laboratories Division of Product Quality Research in Office of Pharmaceutical Quality, and provides hands-on training to reviewers on various topics, including continuous manufacturing, PAT implementations for process control, 3D printing, and pre-formulation consideration for development of oral drug products. And Dr. Zidan is the government liaison of several USP committees and co-leads several manufacturing research working groups at CDER. And Dr. Zidan chaired various events at FDA internationally, and he is also an editorial member of several pharmaceutical journals and co-authored many articles and book chapters of manufacturing and characterization of complex drug products. But with that, I'd now like to hand over to our first speaker today. So welcome, Dr. Ahmed.
Thank you, Marcus, for the introduction and the invitation to share the webinar. So in our talk today, we are going to discuss about design space for continuous manufacturing using weight granulation for production of extended release tablet. We are going to go over some opportunities and challenges. And I'm delighted to share the session with Owen and Rodolfo, which are partner with us in continuous manufacturing research. So let's make around my presentation with this disclaimer that this presentation reflects my views and shouldn't be construed to represent FDA's views or policies. So, everyone deserves confidence in his next dose of the medicine. And this is the mission of our Office of Pharmaceutical Quality, is to assure availability, safety, and efficacy of every dose of the medication. And this is a slogan of the work that we are doing in FDA about pharmaceutical quality. So in this presentation, I'm going to cover continuous weight granulation and how we can fit it into a continuous integrated line for production of extended release tablet. And I'm going through one of the FDA case studies about using weight granulation, twin screw granulation in particular, to produce extended release tablets that has unique formulation. And I'm going to conclude with our learning, learning points from the study and how we can use the outcome of the study in order to pave the way for wet granulation into wet continuous granulation into the arena of Industry 4.0. So, like the selection of a tableting procedure depending mainly on the characteristics of the powder blend and the intermediate product, which is the granules, for example. And the most important parameters that determine the selection of the upstream processing is the flowability and compressibility. And over this plot, we can see that we can classify materials into low and high flowability and compressibility, and aligning these processes into it. And of course, the most easiest way to produce tablets is by direct compression, and this is for materials that's highly flowable and compressible. And when flowability is an issue, however, compressibility is acceptable, then we can go for dry granulation. But when compressibility is an issue for upstream tablet production, then wet granulation would be the choice here, where it can improve the compressibility by adding a binder liquid to the powder. And of course, because it will produce granules, so flowability is improved in both cases. And interestingly, that most of the products in the market is produced by wet granulation. 60% of these produced by wet granulation, either immediate release or extended release tablet. However, only 10% is produced by direct compression, and the 30% is produced by dry granulation. So this is focus on the importance of wet granulation and how we can fit it into a continuous production or continuous manufacturing line. So wet granulation is a process of enlargement of the particles into multi-unit agglomerates with the help of addition of binder solution. It adds some advantage to the formulation, so it improves the particle size distribution and avoids segregation. And of course, it improves the flow characteristics. And it might be a tool to densify the powder particles. So it may be a basis for for extended release by increasing the density of the powder. And also it has also a safety, I would say, advantage by limiting the level of dust during manufacturing. Traditionally, twin screw wet granulation is a process that's used for wet granulation, where the process here has two screws that's fitting together into a barrel, and it is keep moving. So the powder is fed using Lawson weight feeder, and the liquid binder is then fed into the screws, where a complete kneading and mixing process is happening by the screws. So compared to the batch wet granulation, using high shear granulation. Twin screw wet granulation has a short processing time, so it is in seconds compared to minutes or even more for batch granulation. And it has intimate kneading process, so we can have more efficient kneading and distribution of the binder liquid, and it has a potential for high drug load formulation, and also it has the capability of integrating a lot of PET tools to it. So this can be easily, I would say, configured for continuous manufacturing line. So depending on the formulation, there is a lot of process parameter in twin screw wet granulation that may control the performance of the granules produced to it. For example, the powder feeding rate and how it is filled into the barrel, and the screw speed, as well as liquid to solid ratio. And of course, one of the most important is the screw configuration. So this screw can be assembled with different regions of kneading and conveying and sizing elements, even to produce a unimodal particle size distribution. And this is mainly depending on the amount of shear that is applied to the powder, as well as the mixing efficiency of the binder liquid to produce granules of desirable characteristics. So in order to understand more how these parameters may fit into the production of extended release product, let's go beyond the mechanism of granulation here. So we know that as the liquid is added to the granules, then liquids start to form some sort of bridges in between the particles. So it goes from a pendular phase to funicular phase. And then as the liquid addition increases, then it starts to go around the particles. So it has a capillary phase. And with the increase in liquid addition, it will saturate all the particle surface to form this low density droplets. And of course, during the twin screw granulation, shear is applied by the kneading zones of the screws. So by increasing the energy input, then high density droplets and granules may be obtained. And in order to understand the regime map for this granulation mechanism, we can correlate the deformation parameter. The de- I'm sorry, I
Sorry
believe.
about that. 1 second, Ahmed. So there's some background noise coming from one of our microphones. So
Yeah, I'm sorry about this.
yeah, it's okay. We'll just double check that microphone's on mute, apart from Ahmed. And sorry, let me hand back to you, Ahmed.
Sure.
Okay, over to you.
Thank you. Yeah. So in order to understand the mechanism of granulation and how it can fit in designing the space for the granulation, this plot here between the deformation factor, which is a parameter correlating the stress applied to the granules to the, I would say, breaking strength of the granules, against the binder viscosity and liquid to solid ratio. Usually, the deformation factor is inversely, I would say, proportional to the breaking strength of the granules. So, meaning that lower beta here is indicating a strong granule system that is high density and may withstand the deformation during compaction. So, as you can see here, the granules can be divided into 4 zones. And this is depending on amount of shear applied as well as the amount of liquid and binder viscosity added to the powder during granulation. So the first area is underweighted powders where the binder may not be enough to produce bridges and to produce coherent granules. And then by adding more binder liquids, then it will go to a nuclei where some, I would say, seeds of granulation start to form. And by applying more shear, it will go to a cramp and then form a nice granules after that. So by increasing more binder liquids, then it will go to overweighted powders. Or overweighted granules, where in this case it may be affected mostly by I would say, the formulation ingredient, specifically if it has hydrophilic polymers, then we may have high level of swelling that's happening, which is one of the failure mode. So as you can see here, the p-value is inversely proportional with the powder feeding rate. So by increasing the powder feeding rate at the same liquid to solid ratio, then we may have stronger granules here. And as we increasing the powder, the binder amount, then the beta value decrease for the same powder feeding rate. So understanding this one, it may be applied easily to immediate release dosage forms or immediate release formulation, where mostly the formulation is formed of a bulking agent with a limited amount of hydrophilic polymer there. So for extended release formulation, when you have a large amount of hydrophilic polymer that may swell in the granulation tunnel, in this case, I think understanding more about the process parameter to identify the proper or optimum design space is the key in this case. So for extended release formulation, let me just highlight that for now, up to now, there is no any matrix-based extended release tablets produced by continuous manufacturing lines have been approved yet. And most of the matrix-based continuous granulation is used mainly to produce immediate release tablets, in addition to like a lot of direct compression produced by continuous line. So there is a need to leverage our, I would say, lessons and experience that's gained during continuous manufacturing by direct compression into continuous manufacturing by wet granulation for extended release dosage forms. And of course, the interaction between the process and formulation parameter are the critical part for controlling the degree of swelling and the final characteristics of the granules. And this will lead, of course, to avoid a lot of the failure modes that had been observed during wet granulation of extended release formulation. For example, premature consolidation, where the powder start to compress at the first kneading zone, even before addition of the liquid binder, and mainly increasing the torque and amperage of the granulator is observed. And sometimes we have seen excessive swelling, and this happens when large amount of the binder liquid is added to the powder at high liquid to solid ratio. So this leads swelling of the polymer, and the amount of shear was not enough to break it down even. So it will start to coat all the screw, as we can see. And of course, this is one of the failure mode. And in another case, we have seen some poor cohesion where the liquid binder is not evenly distributed over the powder. And then we have a mixture of dry granulated and wet granulated particles produced. And you can see the shape of it here. And of course, the fluctuation of the flow and steady state is always observed in this case. And some other failure mode had been observed where excessive swelling is happening. However, the shear that's applied by the kneading zone was enough to break down this film of the swelled particles to form some sort of spaghetti-shaped granules. And indeed, optimize the design space by finding the reasonable liquid to solid ratio along with the shear that's applied by the screw configuration can help avoid these failures. And G-formed granules is always produced. So determination of the design space can avoid all of these failure modes. And of course, this will facilitate the adoption of continuous granulation by twin screw granulation of extended release formulation. And this is may pave the road for introduction into Industry 4.0 arena. So we can go over one of the case studies that FDA Labs has to define the design space for this continuous wet granulation process. And the main objective of this study was to understand the effect of screw design and operating parameters on the residence time distribution characteristics, as well as the performance characteristics of the granules and the finished extended release product. And of course, identifying the optimized design space for a specific or a particular extended release formulation of metoprolol succinate as a model drug in this formulation. So let me highlight a little bit about the formulation characteristics. So as I just mentioned that for most of the immediate release formulation, the formulation has large amount of a bulking agent and a very low concentration of hydrophilic polymer. And the small volume in the granulation tunnel does not allow to increase this amount of the hydrophilic polymer due to the large swelling volume attained upon hydration. So for this formulation that has metoprolol succinate and it is a readily soluble material, it can break down any matrix that's formed by swelling of hydrophilic polymers in the formulation. And this is necessitates the use of large amount of hydrophilic polymers. and even coating the granules using extragranular hydrophilic matrix after the granulation process. So this formulation unique in the amount of hydrophilic polymers in it. So it has more than 50% of hydrophilic polymers, in addition to an extragranular fraction of hydrophilic polymers, and using BVB here in the binder aqueous liquids. So the experimental plan included mixing all ingredient in a batch mode and then feeding it using loss in weight feeder to the granulation process, and adding the liquid binder here, and the granule produced is then dried in a batch mode, followed by milling to produce the granules, and then the granules of specific yield fraction is then sieved and then mix it with extragranular fraction and lubricated for tablet press. And this tablet is then characterized. So granules produced were characterized for particle size distribution and the many bulk properties like density, compressibility, cohesion, permeability, and flow factor. And the tablet produced were characterized for breaking strength and dissolution. And of course, the process was monitored for mean residence time using both a mechanistic and trace injection, bolus injection technique. And let me highlight here that the old production line was not continuous, but the integrated continuous batch operation where the feeding and granulation and then the semi-continuous drying phase. However, all other unit operation was mainly on a batch process, and the main focus was to study how we can fit the with twin screw granulation into a continuous line. So we have used definitive screening design of experiment by varying two factors from the process parameter, which is the screw speed that was ranged from 500 to 800, and the powder feeding rate that ranged from 5 to 8 kilogram per hour. And only two significant screw configuration parameter, which is the number of kneading element per zone. And this was ranged from 2 to 4 kneading element, and the number of sizing element at the end of the screws ranged from 1 sizing element to 3 sizing element. So all other factors were kept the same for all the granulation run, and the definitive screening design resulted in 18 granulation runs that was used in this study. So for studying the mean residence time and residence time distribution parameter, we have used a semi-mechanistic model. This semi-mechanistic model was relying on determining the volumetric hold-up factor, which is a function of the flow rate of the powder in the granulation tunnel and the available volume that's available for the granulation. So by dividing the hold factor by the flow rate times the displacement ratio by one conveying element of one lead length, then we can determine the mean residence time. So in this equation, you can see that there is a mini tuning model-dependent constant, B1 up to B6, and these constants were used to calculate the mean residence time and Bicklet number, as well as variance and mean-centered variance for distribution of granules characteristic within the granulation tunnel. And these constants were calculated or adapted from previous studies that use the same screw configuration, but it was using different formulations than ours. So this mechanistic model, it was only concerned with the process parameter and screw configuration. However, it does not consider the formulation parameter. And for this model data was validating using tracer bolus injection method, where a certain orange dose of 10 milligram or 11 milligram was injected into the granulation along with the powder, and then, as you can see, by injecting the sodden orange, the color of the granules is changing from white to canary yellow color, And then by using an online camera, we can monitor the change in the color intensity by breaking down the video into images per unit time, and then determining the color intensity, RGB colors, and then plotting the color distribution per unit time. And this is how we calculated the mean residence time and other residence time distribution parameters. So the data calculated from the mechanistic model showed that screw speed and number of sizing element was negatively affecting residence time distribution, and the feeding rate and the kneading element positively affecting the mean residence time. And the highest mean residence time were obtained at a low screw speed of 500 RPM and higher feeding rate. And of course, like increasing the powder feeding rate and decreasing the screw speed is increasing the volumetric hold-up factor. And this was observed also by decreasing the number of kneading elements. So to validate this mean residence time distribution or residence time distribution parameters, these data were compared to the data collected from the tracer bolus injection. And for this validation, we have used only 3 granulation run, G1, G2, and G10. And for all of these granulation runs, they have the same screw configuration. So only for the same, for the 3 runs, 2 kneading element was used and 1 sizing element was used. And for G1 here, it was using low screw speed and high powder feeding rate. That's why it has more, I would say, broad residence time distribution, as well as it has more lag time. And for G2 here, it has high screw speed and high feed rate. That's why it is intermediate. And for G10, it has high screw speed and low feeding rate. So the lag time was much less and the residence time distribution was more narrower. So comparing the data from the bolus injection to the mechanistic model that we have used, we find that so the actual data here is the one we obtained from the bolus injection. Tracer bolus injection, and the predicted is the one that's obtained from the mechanistic model. So we can see that the lag time, mean residence time, and the covariance and the hold-up factor, they were all correlating with the feeding rate and decreasing the screw speed. And the main differences that was observed between the actual and predicted for the variance and mean residence time, this can be attributed mainly due to the fact that the semi-mechanistic model was trained with mean residence time measurement acquired on a conveyor belt after the granulator. However, in our study here using the tracer injection, the data was acquired just at the exit of the twin screw granulation tunnel. So the semi-mechanistic model also didn't consider the interparticulate interaction and the formulation attribute. For example, the interaction between the binder droplets and the powder particles. So that's why there's some variations that's obtained. However, both techniques, the mechanistic model and the tracer injection, they were able to detect the effect of the investigated process parameters on the powder characteristics. And to further understand the effect of these parameters on the particle size distribution, particle size distribution parameter were determined in terms of D10, D50, D90, the 3-tier particle size distribution parameter. And also we calculated amount of fines and amount of oversize and the yield percentage, which is amount of granules in between 180 micrometer up to 850 micrometer. And this is the one that's used for further tableting. So generally, we found that the screw speed has a positive effect on the particle size distribution. So it increased the BSD parameters and it decreased the amount of fines and increased percentage yield and increased the oversize. And indeed, at the same powder feeding rate and the screw configuration, increasing the screw speed lead to incomplete filling of the screw tunnel and leads to shorter mean residence time. And this has led to less friction and collision between the granules, which is allowing consolidation and the granules to grow to increase the particle size distribution. So the highest particle size was obtained by increasing the screw speed and by decreasing the powder feeding rate. So the powder feeding rate has a negative effect on the particle size distribution, and it increases the fine, decrease the oversize, and also decrease the yield. So nonetheless, increasing the powder feeding rate, it increases the particle-to-particle friction as well as particle-to-wall friction, and leads to more attrition of the granules, making it smaller, and a smaller particle distribution parameter was obtained. So we have seen here in this response plot that percent of fines increased at higher powder feeding rate and when less number of kneading element was used per each kneading zone. And for the effect of kneading element, it was also significant and similar to the effect of screw speed on the particle size distribution. So it leads to decreasing amount of fine and decreasing the yield and increasing the amount of oversize. And of course, when the screw speed and powder feeding rate were kept constant, increasing the number of kneading element, increase the efficiency of distribution of the liquid binder, so it improves the mixing and it allows the granules to grow to form more dense granules. So the amount of oversized granules in this case was high when the number of kneading element increased to 4 kneading element per zone, and when we decrease the number of sizing element, that increase the breakdown of the granules at the end of the screw tunnel. So the bulk characteristics of the granules were also studied. So the flow and cohesion and of the granules were determined using the shear cell. And by constructing the yield locus, we found that the flow factor and cohesion of all granules were inversely proportional. So granulation run that has high flow factor, it has lower cohesion and vice versa. And for the effect of the parameter, we found that only the kneading element showed a positive significant effect on the cohesion of the granules. And the interesting results here, we found that the screw speed, I would say, abolish the effect of kneading element by the interaction with the kneading element effect to make it inversely proportional with the powder cohesion. And indeed, the reason for this, as you can see in these pictures, increasing the number of leading element, increase the iterations applied and the shear applied to the granules to form irregular shaped granules, and these interlock together to negatively affect the flow of the material of the granules after that. So the density of the granules and compressibility were also calculated or measured using the shear cells at different applied stresses. And the data showed that the density of the granules was inversely proportional to its compressibility. And in general, the screw speed was positively affecting density and negatively affecting the compressibility. Powder feeding rate was negatively affecting the density and increasing the compressibility. And the kneading element and sizing element were also decreasing the density. However, it was increasing the compressibility of the powder. So we can see here that the density of the granules were mainly were affecting the compressibility, and the highest density of the powder of the granules were obtained when large screws or high screw speed was used, 800 RPM, and by decreasing the amount of fines. So this allowed the particles to consolidate more during compressibility testing. So permeability of the granules were also tested using the shear cell by measuring the pressure drop across the powder bed by applying a normal stress. And we found that the permeability decreased by increasing the feeding rate and increasing the kneading element. And the highest permeability of the granules were obtained when the condition bulk density and the compressibility were at low values. And of course, materials with high compressibility tend to form a tightly packed mass, which is increasing the pressure drop across the bulk granules bed. And this is, of course, decreasing their permeability factor. So to further understand the effect of these process parameters on the tablet characteristics, breaking strength was studied, and for all the granulation run, it resulted in production of tablets of close breaking strength values. So all the granulation runs showed tablets of breaking strength range from 4 kilobond up to 6.2 kilobond. And the screw speed was the most significant factor affecting the tablet hardness in a positive fashion. And the sizing element was non-significantly negatively affecting the breaking strength. And the most interesting is that the sizing element, despite its effect was non-significant, but interacted with the feeding rate, and it predominate for its effect on the breaking strength. So the interaction of sizing element with the feeding rate, it was overall negatively affecting the breaking strength. And this interaction can be seen here on this interaction plot. So the effect of sizing element can be only seen at higher value of the feeding rate. However, at lower feeding rate, the effect of sizing element was demolished. So this behavior can be linked to the increase of the breakup of large granules produced at the high screw speed during compaction. So to further understand the effect of these process parameter on the tablet dissolution characteristics, so we have measured the dissolution rate using the USP method in the monograph for metoprolol extended release tablet, and we have used the same acceptance criteria listed in the monograph. So as we can see that the initial time point and the final time point of the solution were like slightly affected by the process parameters. However, these process parameters were mainly affecting the intermediate values of the solution, which of course is affecting availability and systemic exposure for the drug during the clinical testing. So to understand the effect of this process parameter on the dissolution, we found that screw speed and the kneading element were negatively affecting drug dissolution, and their effect were significant. However, the feeding rate and sizing element, their effect were not significant, and the feeding rate were positively affecting drug dissolution. So of course, increasing the screw speed leads to increasing the breaking strength, and it increases the drug release by producing more dense tablets. And increasing the number of kneading element, it increases the number of shear and compressive forces that is applied to the granules or the powder during granulation, and leads to produce of dense granules to decrease the drug release. And the slowest drug release was obtained at a higher value of screw speed and the higher value of feeding rate of the powder. So as we can see, over all of these data collected on the intermediate and final drug product, the design space for wet granulation can be easily optimized to produce a granules and tablet of desired characteristics. And some of the challenges that was observed, it was coming from a formulation. So some of these challenges was related to like the use of excipients of different functionality. For example, water soluble and swellable and hydrophilic excipients can affect this design space, and this may necessitate the use of co-processed excipients. But if the blend uniformity is an issue of this co-processed excipient, then more intimate mixing may be needed. And the overall, I would say, bulk properties of the blend affecting also this design space, API loading and attributes, for example, particle size distribution, size and cohesiveness, as well as the swelling space to obtain a steady state processing. And most of the swelling process may be overcome by using organic based binder liquid. However, it may have some safety concern during a large scale manufacturing. Some other challenges were related to the equipment design, where, for example, if the equipment design is mainly for a certain formulation, or how the wet granules is affecting the downstream processing. For example, the drying mechanism, is it fully continuous or semi-continuous, or will it be in batch mode? So improving our process understanding from the formulation and equipment design, it will facilitate introduction of wet granulation of extended release product into Industry 4.0 arena, where we can implement PET and artificial intelligence and machine learning in development a full control strategy to the granulation line. So it is feasible and it can be applied for certain formulation considering all of these challenges. So at the end of my presentation, I want to acknowledge the team here in FDA and our partners from the emerging technology team. I want to acknowledge Dr. Sierra Vega for his efforts during this work and industry, our academic partners. So this is my presentation. Thank you for listening and attention to the presentation. And then we can go again to Marcus for the second presentation. And for follow-up questions, we can take it at the end. Thank you.
Thank you very much, Ahmed. And I echo his last comment as well. If you have any questions for Ahmed, then please do feel free to send in as many questions as you like via the questions tab. You can find us directly below your webinar screen. And we look forward to going through your questions later during our live Q&A session. Okay, great. Now, please allow me to introduce our next speaker. So, Owen Rehrauer is the president of Sentronic US Corp, and in this role, he is responsible for service, sales, and technical support of Sentronic instruments in the United States. Additionally, he is providing global support within Sentronic for for chemometrics applications and continuous manufacturing implementations. And Owen holds a PhD in chemistry from Purdue University and joined Sentronic in 2022. And prior to working at Sentronic, he worked as a PAT scientist at Vertex Pharmaceuticals from 2016 to 2019 until starting the PAT Part NIR LLC consultancy business. Okay, and with that, let me hand over to our next presenter. So welcome, Owen. Oh, just going to stop you there, Owen. You are on mute. Let me unmute you quickly. Go ahead. You're good to go.
Sorry about that. Thank you, Marcus, and thank you for the opportunity to talk. And now hopefully people can hear me. So just a quick overview of Sentronic for folks that have not encountered us before. Sentronic is a company that's been in business for 31 years now. I, as Marcus mentioned, joined 2 years ago when we opened up the American subsidiary. Sentronic has been focused on spectroscopic or sensor-based process solutions throughout its lifetime. Two major divisions. One is gas sensors, which is not the area I work in. The other is NIR-PAT, with a strong focus on oral solid dose pharmaceutical manufacturing. We also do some chemical ID for German admixing in the pharmacy market. Most of my day-to-day, though, is focused on what we can provide for PAT solutions for pharmaceutical manufacturing. Here's just a quick summary of different solutions we have for different unit operations within manufacturing. But having said that, let's get on to the talk. The subject of today's talk is something I spend a lot of my time thinking about, and my commercial customers and customers moving to commercial spend a lot of time thinking about, and that's PAT reliability. This is not a subject that gets a lot of technical talks in venues like this because from a narrative perspective, it's not as interesting as some other items. Namely, if you develop a reliable solution, nothing out of the ordinary happens. That makes for something of a boring story. But having worked in that environment myself and working with customers who are very candid with their opinions on it, sometimes boring is very desirable. Going home on time is very nice. Having a plan execute according to plan is very nice from a business planning perspective. So reliability is important. And as we see PAT be more broadly integrated into the pharmaceutical manufacturing environment, I think having PAT reliability should become more of a common discussion. So today I'm going to talk about how we approach that problem. It is only one solution. There's a number of different ways one could do this, and just go through our experience and our current generation of instruments as a bit of a case study on ways to approach this problem. But let's set this up. So why are we discussing this? Well, I'm going to use the term of art the government uses, smart manufacturing. I know the next speaker may have some strong opinions on the term advanced manufacturing, but we're seeing more and more of this spread across the US and Western Europe, and England as well if we don't count them in Europe. This is occurring because of reshoring for a number of different reasons, but supply chain reliability tends to be a non-trivial part of that. And when these environments reshore, when these manufacturing operations reshore, it tends to be using a lot of automation to enhance productivity given the higher labor cost of these markets. So the way we see that integrated tends to be something like smart manufacturing. And in the pharmaceutical industry, one of the more common, one of the more emerging types of smart manufacturing is continuous manufacturing. So how does PAT reliability impact continuous manufacturing? There's a really interesting paper out by the folks at the University of Tokyo and some folks from Daiichi Sankyo that have put together a decision support matrix. This decision support matrix is available in the linked paper here. It's an open access paper, so anybody can go there and get this information. And what this decision support matrix does is lay out the case for when it makes more sense to batch manufacture or manufacture continuously. So when we see continuous manufacturing operations, one assumes there's been an analysis done. It's been determined that for a given product or a given family of products, continuous manufacturing is more beneficial economically to use than batch manufacturing. So there's a number of different variables, quite a large number, that influence this decision. You can end up with a contour map like the one I've shown here from the paper, and there's a number of different factors that affect this. A lot of these are going to be process and material specific. So for a given drug, the API costs, commercial price per tablet, et cetera, impact is. But some of these are fixed, some of these are operational, and PAT falls into that bin. While it's certainly not the most important factor here, that tends to be more product-specific materials. Of the non-product specific properties, PAT operation, PAT uptime is a non-trivial part of this decision. In other words, if we want to see more continuous manufacturing happen, when we look at a plot like the contour map shown here, we'd like more of that map to be pink, where it's continuous beneficial, than blue, where it's batch beneficial. And having highly reliable PAT requiring a smaller number of work hours to keep the PAT operational, and if the PAT is critical for continuous manufacturing, which it often can be, if PAT downtime occurs, then the whole operation doesn't happen, which has a very large impact on the reliability of the CM operation itself. So anything we can do to decrease the mean time between failures for the PAT itself becomes of the utmost importance. So what are some reliability challenges for PAT? Where is PAT as a technology in the technology lifecycle development process? Well, PAT has been shown to be technically feasible for pharmaceuticals for decades. We have papers from the 90s demonstrating its usage and its applicability to the pharmaceutical manufacturing environment. It's been used for almost 10 years.
I'm just going to stop you very quickly. Your slides have just gone off screen. So just check your share screen and let's get it back up again.
Sorry about that.
It's okay.
Apologies, folks. Is that better?
It's still on full screen mode for our cameras.
Oh, shucks. Oh, that would do it. Apologies for the technical difficulties, folks. Is that better?
That's better. Yeah.
Awesome. Sorry. So where are we in the PAT technology lifecycle? We know that PAT is technically feasible. It's been used in commercial manufacturing for almost 10 years now. Early PAT was developed using laboratory instruments. We look at some of the early work from places like Pfizer, where they used a laboratory instrument, took a process probe, integrated it into a BIM blender through the arm of the BIM blender, developed more fit for purpose PAT solutions for BIM blending, things that tended to be wireless and were more robust to the manufacturing environment. And I want to call out the distinction between a laboratory instrument that's been adopted for the process environment and a true process analyzer. So how does a laboratory instrument differ from a process instrument? Lots of different ways, and some of this is a bit common sense, common knowledge, but in brief, laboratory instruments are designed to work in a laboratory, as the name might imply, and everything else being equal, they tend to focus on having as high of performance as possible, performance being measured, for example, in signal to noise, spectral resolution, etc., relative to the cost of the instrument. Measurement time tends to be whatever is necessary to achieve its performance needs, and it's designed for use like with trained operators. In contrast, process instrument needs performance that's fit for purpose for their application, but robustness really becomes key, whether that's robustness to environmental interference parameters. A tablet press is a bit more shaky than what you typically find on a laboratory bench. So being robust to environmental changes like temperature changes in a process environment, being robust to vibrational changes, all things that are important. Being robust to routine handling by operators that maybe aren't trained PhD scientists tends to be a bit important. And I don't want to forget to mention measurement time, which also needs to be fit for purpose. If we're trying to control a process in a timely way, we need to be able to make measurements that are fit for purpose for that time scale of control. In other words, if I had a PAT system that was highly, highly accurate, but it took me 20 minutes to measure, that may not be so great for controlling a continuous manufacturing operation at the tablet press. So just some differences between the two classes of instruments. And if we want to industrialize PAT, we want to make sure it's fit for the needs of continuous manufacturing to do this in a commercial way, not just demonstrate feasibility, but really be ready for use 24/7 as some of our customers will use it. We need to, at the design stage of the PAT instrument, consider both reliability as well as spectroscopic performance considerations. I can tell you from personal experience, reliability doesn't happen by accident. This is something that we need to intentionally consider and design different aspects of the instrument to ensure that it is there. So for continuous manufacturing, what are some things PAT should do based upon what we've heard from our customers and their user requirements? It needs to be fit for purpose, so at a minimum conform to the regulatory guidances or just the general technical guidances shown here. Needs to be able to make measurements that are fast enough for timely control, needs to be robust and make robust measurements 24/7. So measurement at the start of the day looks the same as the measurement at the end of the day if the material is the same. Provide real-time alarms if the system's not working to specification, so people know not to trust the output. Have probes that make sense for the process and can interface with the process and get representative sampling, and be robust to routine handling. So I'll walk through our approach to solving this problem. I do want to call out this is not the only way one could approach this problem. And if the spectroscopy and the process under review was different, for example, if this was a bioreactor being controlled by Raman, the sorts of decisions being made would be pretty significantly different. So I'm not saying this is the only way or the correct way to do this, but it's the approach we've taken, and we'll talk through what the benefits of this approach are. So when designing a spectroscopic system, one of the first decisions you should make is probably spectrometer. Lots of different spectrometers are available for NIR spectroscopy systems. We tend to favor diode array-based solutions, and there's a few reasons for that. Relative to something like a Fourier transform spectrometer or a DMD-based system using Hadamard scans, there's no scanning of different material as a function of time. When you have a flowing material flowing past a process probe, measuring all the wavelengths of light at the same time has some real benefits and presents no risk of spectral artifacts from the scanning process itself. I'll talk about this in the coming slides, what the risk of spectral artifacts might be for a scanning system and what those might look like. Diode array systems allow very, very fast measurements, which is a nice thing to have for fast process control. I will note here, we tend to scan on the time scale or measure on the time scale of 8 milliseconds to 10 milliseconds. I keep catching myself saying scans. We really prefer the term coads. That would be the individual 8 millisecond or 10 millisecond measurement. Then we will average a number of those very fast measurements together to get something like an average spectrum on the time scale of maybe every half second or 1 second. You may be more comfortable calling those scans from using a Fourier transform. I will note there is actual no scanning here, so we tend to prefer the term coads. These types of systems are very easy to design, so they conform to the aforementioned USB EP requirements, and they contain no moving parts. This is a nice advantage in terms of vibrational and temperature robustness. So if there's vibrations in the environment or there's temperature change in the environment, everything else being equal, this is a fairly robust system, although obviously it's not bulletproof. Please don't hit the system with a hammer. You will definitely see some changes there. So let's talk a little bit about spectral artifacts. I mentioned before, we're measuring all the wavelengths of light at the same time on a very fast time scale. Something that's scanning through a spectrum will measure different wavelengths of light, different combinations of wavelengths of light over a long period of scans. And that can take on the order of 100 milliseconds. Where that presents a risk is if sample presentation to your process probe changes on the time scale of the scans. One way that can happen is if material is closer or further away from your measurement spot. For example, if you're measuring in a chute, you can have more or less material presented to your probe. If you're measuring in the feed frame of a tablet press, you may have changing heights of the bed that you're measuring, particularly if you don't have a fully filled tablet press, and that can present significant changes in the reflectivity of the material under review. So we've done a quick in silico experiment here where we change the reflectivity of the spectrum of lactose. So we normalize it and change the spectral response based upon the reflectivity one might see over 100 milliseconds. We then simulated what that would look like if we measured with a diode array system every 10 milliseconds and simulated what that would look like for a Fourier transform measurement over the 100 milliseconds. In the case of a diode array spectrometer, we see that we have a constant baseline offset, but otherwise the peak locations are all the same, and there's no new peaks coming or going over these measurements. In contrast, for the simulated FT spectrum, I've put here in blue the reference spectrum of lactose we use for the simulation, and in red, the Fourier transform recovered spectrum with the reflectivity change. You can see there are significant changes to the peaks of that spectrum. This, of course, is a risk. Now, one could argue that you could engineer ways to ensure this does not happen. I think that's absolutely the case, but we have made the decision to go with diode array just to avoid this as a possibility and not need to engineer a solution to the problem. So we've picked our diode array spectrometer as the way to go. There are some reliability challenges with this, though. So there's upsides and downsides. One of the challenges is x-axis drift. We're more likely to see drift in our x-axis on a diode array system, everything else being equal, relative to a Fourier transform system. This drift and other challenges with x-axes can result in some challenges transferring methods from instrument to instrument without a method update relative to an FT system. Intensity drift is also a bit more likely than an FT system, and the resolution is not as high. Although in the case of resolution, I'll note that the resolution of a diode array system often remains fit for purpose for PAT application. So this is a case where we have a process instrument, we need to be fit for purpose. The resolution is going to meet that bar, so we're not going to stress about it. But the remaining three items are things we've had to engineer solutions for to ensure reliability, ensure that the instrument remains fit for purpose. So how do we address these three areas? In the case of x-axis drift, we've actually incorporated an internal wavelength standard. We use a Fabry-Perot filter. For those unfamiliar, this is two thin pieces of film with an air gap between them. When we move this into the optical path of our system, we will actually see something that looks like a sine wave across our spectrum. and we can characterize where the minima and maxima of this sine wave pattern are. During factory testing, when we calibrate our system, we calibrate using NIST traceable standards, and then we immediately. So we then have a relationship between where these peak locations are when the system is well characterized. And if we make a measurement at any given time, we can say, okay, we found the peaks here, and we can correct them to be where they should, minimize the error of that fit when the system was well calibrated. This helps lock in the peak locations. So let's challenge the use of this. One cause of x-axis drift in diode array spectrometers is changing temperature in the environment. So if we put one of our systems in a temperature control chamber and vary the temperature intentionally while using a correction using this internal wavelength standard every 10 or 15 minutes, what does that do to the x-axis? So if we, say, measured a NIST 1920 standard on our probe, look for the peak locations of several peaks, do those peak locations drift as a function of temperature? And here I'd argue we remain fairly constant. And outside of the case of the 1935.5 peak, which does have a little bit more variability, we don't see large changes in the peak location, which is nice. This is a nice stress test. Most of the time, our systems are not seeing 15 degrees C changes over a handful of hours. That would be very unusual. But even if we did, using this technology, our x-axis remains locked in. So we believe this addresses the concern about x-axis drift. But let's talk about instrument-to-instrument robustness. So it could be a major added headache in a commercial environment if we did all our model development on one instrument and that instrument experienced a catastrophic failure. If we had to put in a second like-for-like instrument in, it would be really excellent if we had a high degree of faith that after validation, we did not have to do a method or model update for the second system. This can be very challenging with diode array systems, and we've developed a solution to, as best as we can, and we of course can't give a guarantee, but I think we have some interesting case studies demonstrating that this generally is true for a typical application, can give customers some degree of faith that one of our systems will look like the second system, will look like the third system. So how do we achieve this? We use a procedure called factory averaging. What we used to do many, many moons ago before I joined the company was when we calibrated our system, measured a reference standard, typically a 1920, find a number of peak locations, and then on an instrument by instrument basis, calibrated to minimize the error of fit to those locations. The challenge with that was that instrument one would find one fit, instrument two would find another fit. They didn't know anything about each other. And we could have wildly different responses on an instrument to instrument basis, even if those were the best fits for those two instruments. Around 2017, we changed to a new factory averaging procedure. When we do this, we take the second derivative spectrum of the 1920. We find over 60 peaks in the second derivative space, and we characterize those. We take a fingerprint. We've done this for a large number of systems at our factory, and we found the average response of that large number of systems. We then minimize the error of fit for these 60 plus peaks to that factory average on every system. We still check to make sure we would pass our USP EP testing criteria, which is not more than plus or minus 1 nanometer off of the reference peaks. We consistently pass that. But what this does is it makes our systems look more like each other. So a method developed on one system will be, everything else being equal, highly likely to still work on a second system. That's a lot of words. Let's actually see what this does in practice. So we had a customer bring 6 systems to our factory. They had an APAP blend, so a real sample. This blend was measured on these 6 different systems. We made a PCA model for the spectra of the blend from the 6 systems, and you can see on the left graph where those 6 systems fall in PCA space. They fall fairly far apart. We then took those 6 systems, applied our factory averaging technique, measured the blend again after this averaging was done, and then projected those spectrum back on the PCA model developed for the original calibration process. And while they're not in the center, you can see that these samples cluster much closer together, which is another way of saying the spectra look more similar than they used to. that's still a little bit abstract. What does this do in terms of a commercial operation? So the folks at Pfizer a few years ago put out a very interesting paper where they talked through their process of development and how they developed a method and then moved it towards the commercial space. So they're using our systems. This is all in the paper. They developed a method using a benchtop feed frame simulator using one of our systems, and then transferred that method to 2 different manufacturing sites, each with their own NIR system and probe. No model update was required to generate acceptable results from the benchtop method to the methods out in the field. While obviously this is not a guarantee for any given product, there may be products where this does present a challenge, but at least for a fairly typical method, we do have some evidence that our factory averaging is allowing people to build a method on instrument 1, transfer it to instrument 2 without any model update being required. It's a nice feature and reduces potential downtime if there ever was a catastrophic failure of an instrument in a manufacturing environment. So we talked about the x-axis. Let's talk about the y-axis. So why does the intensity drift for a diode array system relative to an FT system? And that comes down to illumination. We're using a tungsten halogen source for illumination as opposed to a laser source, which is commonly used in an FT system. Much like a light bulb or the headlights in a car before LEDs were developed, as a function of time, over the lifetime, you're going to see resistivity changes in the tungsten filament, which is going to cause a decrease in light output. The light gets a little less bright as a function of the number of hours it's been on. We developed these to last for a long time, but still for very, very long measurements. So if your process needs to run for weeks, this decrease in intensity can be non-trivial. It can start affecting the spectrum that you're getting reported. So how do we correct for this decay in intensity or this drift in intensity? I've got a cartoon of our probe shown on the screen now, where the shaft is shown. We have a light source that goes through a quartz rod and then is a free beam out to a sapphire window. That beam can be interrupted using an internal electronically controlled piezo mirror. So we put that into a process, and that is now shining light onto a piece of white reference material that we put inside of our probe. This can be used not as a reference, but as a useful indicator of how intense the light is. So we're getting some information about the intensity of our lamp. And if we measure the intensity at the start of the long measurement and then periodically over the measurement, let's say every day, we can correct for these small drifts of intensity in our absorbance calculation. This whole process takes a very short period of time. So we put this mirror in, make our correction measurement, move the mirror back, continue to measure the process. Oh, shucks, wrong direction. What does this look like in practice? So in the bottom right hand of the screen, we actually have one of our probes. And I'll show you a quick video of this process in practice. So you can see little reflective mirror comes in. That's it. That's how long this correction takes. So we can do this in process without having to pull the probe from process, which may not be obvious, but in practice, pulling the probe from process can also be a little bit of a headache. So if we had to take our probe out to measure an external standard on the probe, that would require handling the probe, cleaning the probe. Any potential risk of containment would have to be managed. That can be non-trivial for customers. And potentially we may have to stop the process. If there's just a hole in the process, that's not really ideal. So by ensuring that we don't have to pull the probe from process, that's a big win for some of our customers. So what does this look like in terms of absorbance? Well, if we measure, in this case, just a white spectrum, so we expect the absorbance to be near zero with the intensity drift correction enabled. We can also back calculate what this would look like without it enabled. And over a 12-hour measurement, you can see with it enabled, stay rock solid near zero. Without it enabled, we're already seeing some drift. That's only 12 hours. You can extrapolate out what that might look like for weeks or many weeks at a time. So it can be quite helpful. These standards, the wavelength standard and the intensity standard, can also be used for system suitability testing. So much like one might do in a chromatography run, where you have fundamental standards which are measured at the beginning of the run, and then every so often you measure a suitability standard just to ensure that your chromatography system is performing to specification. You can trust the results between two different suitability measurements. We can do the same thing for continuous manufacturing. We can periodically assess the internal standards to make sure that the system is performing as we should expect it, and we have a higher degree of faith in the integrity of our results. Running this, you saw how fast that mirror went in and out, takes on the order of 30 seconds to a minute. We get some information about whether or not the system's in good health and can continue running. I'll also note 30 to 60 seconds from an RTD perspective is highly unlikely to have an unusual perturbation to the process that we wouldn't catch had we measured it. So this has been generally accepted as appropriate to do given the benefit to ensuring the PAT is operational. Let's talk a little bit about process and probes too. So we talked a lot about the spectrometer, but the probe and how we measure using the probe is also very important for reliability. Unfortunately, we can't just stick a probe in the side of a pipe and get representative sampling. We need to really put some thought into how we measure and make sure the measurements we're getting are representative, A, of what our reference material might be. So in the case of a tablet press feed frame measurement, we want to make sure that the measurements we're getting from the feed frame are representative of what we'd see in core tablets that we'd use as a reference method. Or upper image here, something like a spider wheel can be used to sample a powder stream to ensure that we get representative sampling of that stream. These different probes all have different requirements in terms of measurement time. So the feed frame of a tablet press has very high velocity material under it. We need to make sure that we can measure quickly enough to ensure timely process controls, typically closer to something like 8 to 10 milliseconds. In contrast, the bottom image here is of a GAA lighthouse probe that we have a probe that is compatible with. In this case, powder is moving relatively slow. We also have 7 spots on our probe to ensure that we're getting a wider area of measurement. And we're able to take a little bit longer when we make this measurement because the material itself is moving in and out of that process zone a little bit slower. So matching the powder velocity to our measurement time tends to be very important, and ensuring that we're getting representative sampling is also important. So a little video here of the spider wheel in action. Unfortunately, you can't see our probe, believe it or not, and you're just going to take it on faith here. It is behind the white powder that is getting measured here. And you can probably see some obvious challenges with this type of measurement. Namely, when the back of those channels on the spider wheel, the back of the little, up notches of metal in front of the probe, the back of that is going to look very different than the spectra of material from our process. It's moving very quickly, as you saw. So you're taking many coads, and only a few of those tend to be from the actual material on the back of those fingers. But that can have a big impact on the average together spectra. Everything else being equal, we'd love to remove those non-processed material coads from our average spectra. So how do we do that? We've developed an outlier rejection algorithm to reject coads, poorly presented material based on some very, very simple math. The most simple way we can do this is just on the absorbance at a fixed wavelength. We tend to pick 1300 nanometers because it's very uncommon that organic materials will have absorbance in this region. So this is really telling us something about offset or about reflectivity. So metal will shoot way, way up for absorbance. We can set a minimum and maximum absorbance that we say this is likely due to a well-presented material. Anything outside of that will reject as an outlier just from our coads. Again, that's a lot of words. What does this do in practice? What does this do to our spectra? On the right, we've got spectra measured using a spider wheel without outlier rejection. So this includes the back of those fingers from the spider wheel. On the right, well, not rigorously the same material, but same experiment, same type of material, we've turned on outlier rejection. Nothing else has changed. I don't know about the folks in the audience, but if I were developing PLS models or whatever control models, I would much rather get the spectra in the blue than the spectra in the red because they look much, much more repeatable. The variance across the spectra is much, much lower. And again, this is just capturing process variation, not variation of the process confounded with the variation of how we make the measurement. Robustness to routine handling is also something that we are very cognizant of and try to design to ensure that normal handling of our systems does not cause damage to the systems, prevent them from being used or create downtime. there's a lot of different ways this can happen. People get very creative. We always are surprised by what we see, but it's hard to put a number on that. So I'm going to use our kind of internal qualitative classifications to describe how we think about this and what drives us. When you have a handling error that causes damage to a system or downtime to a system, there tends to be one of two responses. The first is, yeah, that happens. And the second is, oh, wow. We tend to want to design our systems so as best as we can, we only ever have to say, oh, wow, to a handling error. The image here is one of these classifications of errors. Without naming the client involved, this was one of our blend systems, so not in continuous manufacturing, but I think it's useful here as a story. This was attached to a bin blender, so clamped on well. Unfortunately, an operator didn't fully attach the arm to the bin, so the bin made one rotation and fell with the bin landing on top of our system, bounced, landed on its side. The good news is the system was still fully operational, passed all its validation tests, but as you can see in the image here, its ingress protection was significantly damaged, so replace the housing, send it back to that client, everything worked good. So every now and then mistakes happen. These things can happen, but we would not want this to be something where the system was designed in a way that just routine, normal, non-expert handling caused a problem like this. And this took some time to repair. So try to avoid that sort of situation. So bringing it all together, again, I just want to emphasize this is not prescriptive. This is our solution. We hope it's a useful case study. We hope it gets you to think about how you approach PAT and where reliability factors into your PAT deployment and your control strategy. Ensuring it's robust is really going to be key to ensuring robust adoption of smart manufacturing. It's hard for us to really put a pin on this, put a big number on this. So I'll give another anecdote, what the advantage of this is. And this is an advantage to us as a company, but I think it also speaks to the advantage to our clients. We run a very small company. We support a number of commercial manufacturing continuous operations, which tend to operate 16/5. Those customers tend to have service contracts with a guaranteed on-site response time of 72 hours. We can support that across the country with a staff of 2 people. We haven't had backups. We haven't had 2 calls at the same time. It becomes very, very easy. And I'm going to make a dated reference and say we're not quite at having service engineers that are the Maytag man. We're trying to move in that direction. We want this to be very boring, reliability to be there by design, and for us, to have to show up and do routine service visits or PMs, not have to do emergency response visits. So, having said all that, that's my BioPharma Asia talk. I hope it was interesting and insightful, and I will pass it back over to Marcus. Looking forward to answering your question. Thank you.
Great. Thank you very much, Rodolfo. And if you have any questions, audience members, for Owen or for Ahmed, then please don't hesitate to send in as many questions as you like via the questions tab. You can find us directly below your webinar screen. Okay. And in a moment, we're going to go through our questions. So at this point, I would be introducing Rodolfo, but unfortunately, as far as we know, there's been a technical problem and he's disconnected from the webinar today, and he hasn't rejoined yet. So to keep things going, we're going to move on to our Q&A session. Okay, and joined here by Ahmed and Owen. And our first question to start us off today goes to Ahmed. Okay, so this first question, Ahmed, says here, please describe the criteria used in selection the tracer for monitoring the RTD for the granulation process. Was its formulation or process specific?
Thank you for the question. So actually, selection of the tracer is very critical in continuous granulation, considering the interaction with the formulation ingredient, and sometimes it can interact with granules and the binder liquid chemically, and it can change the swelling profile or the hydration that's happening there. So we have used here Sudan Red, and we have optimized the type of the tracer that we have used, as well as the quantity, so that it does not impart any changes to the granules characteristics. So I would say it is a formulation and process specific, and it has to be optimized because, of course, you need to monitor a process without affecting the process itself, right? So selection of the tracer is a very, very critical process here.
Mm-hmm. Thank you, Ahmed. Before I move on to our next question, Owen, did you have any additional comments to give to this answer?
No, sorry. Reading something else. Apologies for the facial reaction.
Oh, no, understood. No problem at all. And I tend to do this for each question. I just ask if there's anything extra you'd like to add. Okay, so this next question to Owen says, how easy is it to quickly swap out a PAT system once it's been qualified to run in a GMP environment?
Good question. I wish this was truly plug and play, but I don't think anyone that does commercial manufacturing would tell you that the QA group's gonna accept that without a lot of pre-qualification work. So oftentimes, and obviously, I'm not a QA person. It's going to depend on your implementation of GMP. People will need to validate a system as it's put into the process line. Companies that do this commercially will often, but not always, have multiple systems already validated, continually qualified, ready to go. So if one instrument were to go down, they can quickly do a quick PM on the other one and have it ready to go in the line for use. There's a number of different strategies on how to approach this. That tends to be the most common one, though. So unfortunately, we're not at the point where it's just a black box. Okay, I take this part out, put this part in, continue to run, everything's honky-dory. There's a little bit more to it than that, but it's moving in the quicker direction.
Awesome. Great. Thank you, Owen. And before our next question, Ahmed, do you have any additional comments to give to this answer or thoughts?
No, I am second to Owen. I think it is very critical, to use a certain procedure to switch PET from a certain location to another location, or even to switch PET from one technology to another technology. So a full validation scheme and verification to fit for the purpose should be followed, especially in GMP environment. So, yeah, so nothing to add after Owen.
Okay, great. Thanks, Ahmed and Owen. This next question to Owen says here, Owen talked about PAT systems conforming to USP/EP requirements. What are those requirements?
Yeah, great question. It's actually a little bit of a tricky one. So the regulatory guidances that are active at the moment, or not, I keep saying regulatory guidances. Sorry, that's incorrect. The guidances here are USP 1856 and then EP 2.2.40. Those include some different requirements. It depends on which market you're in and which one of these you want to use. To the best of my knowledge, and Ahmed can correct me if I'm wrong here, the USP guidances, at least in the US, don't carry the weight of law. Different territories will have different considerations on that. USP 1856 is less prescriptive than its predecessor, USP 1119, for a process instrument. And I would argue most PAT NIRs are going to fall into that classification. For there, it is prescriptive on photometric linearity, so having a slope between 0.95, 1.05, a intercept of between minus 0.5 and plus 0.5, and wavelength uncertainty, which is 4 calibrated peaks plus or minus 1 nanometer. Photometric noise, they leave that to manufacturer recommendations. At least our implementation, I don't want to speak for other people in this space, we tend to just make that conform to the EP requirements then, which is an RMS calculation. The older USP 1119 did have some segmented noise requirements. We do still have that as an option in our systems. Different people have different requirements there, but high level, it's going to be photometric linearity, wavelength uncertainty, and some degree of photometric noise.
Okay, great. Thank you, Owen. And Ahmed, any additional comments here?
No, actually, this is very, very fair. Yeah, and I believe now there is some harmonization that's happening between different regulatory authority using the ICH Q13 now, which is how to implement control strategy using different PETs, etc. So at the end of the day, I would say it is harmonized specifications all over all the monographs and guidances. So, yeah.
Mm-hmm. Okay, great. Thank you very much, Ahmed and Owen. This next question for Ahmed says here, would you please elaborate on the number of CM products approved for ER formulations processed in TSG?
Yeah, so this interesting question, but for now there is not yet any extended release formulations as processed by tuning screw configuration, tuning screw granulator. And the main reason for this, I showed you, it is very formulation specific, and to implement a control strategy, there is always a need to identify where is the optimum design space and how to implement PET to it. Despite of this, there is many applications that's approved using TG, this for immediate release formulation in a continuous granulation line. But considering that this continuous manufacturing is relatively new, with very few applications that's already approved, and the continuous granulation by direct compression just graduated from the emerging technology program at the FDA just a couple of years ago. So we are expecting more applications to come, and there is also like some application in the pipeline now under review using this application. So I would say it is just undergoing.
Okay, thank you, Ahmed. It looks like yeah. Okay. Before our next question, Owen, any additional comments here?
No. This is a little outside my area of expertise. So
yeah. Understood. Okay, then. This next question to Owen says, does Sentronic have any DMD-based PAT system? If so, for what purpose?
Good question. No. I think the DMD-based systems would be a challenge for a lot of the PAT stuff that we do. It's been a while since I played with the Texas Instruments NIR solution there. I do know that there's been some improvements made, but the time scale would be the thing I'd want to keep in mind. And the risk of spectral artifacts I mentioned earlier in my talk with regard to FT would also be expected to be of concern there. So it's interesting, particularly from a cost perspective, being able to deliver a low-cost device, but not something we're currently doing.
Very good. Thank you, Owen. Rodolfo, thank you for reconnecting. Can you hear me okay? Hold on. It looks like your microphone is on mute. There we go.
Yes, my microphone is very.
There we go. Thank you for rejoining. Sorry for the technical problem on your side, and thank you for reconnecting. We're just in we started the Q&A session because
Sure.
we wasn't sure if you were able
Sure.
to rejoin, but there is still time. Would you still like to present now?
Fine, if it's fine.
Yeah, your connection seems good and strong. Yeah, and Ahmed and Owen, is that okay with you both as well?
Sure.
Yeah. Good. Okay. So let's just set up your share screen. Yeah.
Okay.
If you need me to as well, I can share screen from my side if that's easier.
Okay. Okay, here we go. Let me know if that's working.
Okay.
If that's the presentation. Can you see it?
Nothing's come up yet, no.
Okay.
Okay, this looks good. Yep, we just need to go to full screen mode now.
Okay.
Very good. Yep, we can see this.
Okay, so sorry for I moved over to an area with higher signal. I guess I got disconnected. Nice. Okay, so this is a lot of the work that I'm presenting today is thanks to our research group, and one of the members is now with Ahmed, etc. And so thanks to all the agencies and all the groups and all the collaborators that we've had over the years. We are in Puerto Rico, where we have a lot of pharmaceutical manufacturing in our ecosystem. We export to over 130 countries, and we are very small. That's 161 kilometers by 56 kilometers wide, but with $53 billion in biopharmaceutical exports. And if you want to invest in our ecosystem, these are the people you can contact, my good friends at Invest Puerto Rico. And a few years ago, after working in manufacturing an entire career, I decided to check the definition of manufacturing in an industrial engineering textbook. And so very much what I'm expecting, which is working with materials and transforming those materials through a set of unit operations. But also very interesting, manufacturing adds value. The materials are transformed into products of greater value. And that greater value is not just in terms of dollars. For pharmaceutical products, it also means value in terms of more healthy lives and more fulfilling lives. And so we started talking at NIPT, the National Institute for Pharmaceutical Technology and Education, that groups 18 universities that are very focused on pharmaceutical manufacturing and pharmaceutical engineering. And we started talking, what is advanced pharmaceutical manufacturing? Of course, somebody Googled and said, oh, FDA already defined advanced pharmaceutical manufacturing. Why are you guys wasting your time? Well, yes, there was already a definition for advanced pharmaceutical manufacturing, but this definition is not based on how the pharmaceutical manufacturing might look like. It was just on what is expected. Okay, more quality, less quality problems, less drug shortages, and products in market faster. So how might this advanced pharmaceutical manufacturing look like? Okay, these functions are not the same as its benefits. So this is what we came up, this definition. Advanced pharmaceutical manufacturing is a system that is designed using predictive models where automation minimizes human intervention while enabling closed-loop control and real-time quality assurance, where performance has been optimized to maximize desired process goals, where flexible amounts of product with equivalent attributes can be manufactured, and where equivalent process can be implemented at multiple locations to manufacture products with equivalent quality attributes. This is a very high standard. We realize it. We believe that continuous direct compression already meets this standard. That doesn't mean that other manufacturing does not meet this standard. Will allow other people to justify, whether their process meets this standard or not. But meanwhile, this is our humble definition of what advanced pharmaceutical manufacturing might look like. First, predictively designed, okay, based on process understanding, based on experiments such as those that Ahmed showed today, based on predictive models that will help us guarantee the obtainment of critical quality attributes of the pharmaceutical product, where we do significant evaluation of the physical and chemical properties of excipients and API through an API, where we develop material properties libraries. So these predictive models help us identify less expensive materials that will serve as surrogates during process development, that help us predict the residence time, distribution, material hold up in blenders as a function of material properties and process parameters, that help us develop RTD models for closed loop controls, and help us in the prediction of material behavior and process performance and final product quality. This is also should be optimized, and optimized is when a predictive model is used to select design and operating parameters of the integrating system that maximizes a desired outcome or minimizes undesired outcomes. And so there are now people that collaborators that use flow sheet modeling of the entire system, and this model brings in together the predicted the combined effects of critical material attributes and critical process parameters on process outcomes. And we search for the combination of such parameters to maximize desired outcomes or minimize desired ones. And this is an example of the closed loop flow sheet modeling of the integrated process. But we also need to look at minimizing undesired outcomes. Still, in cleaning validation, unfortunately, we have difficulties in cleaning the systems. And so recently we published an article on trying to simplify the cleaning of continuous manufacturing systems and using the same excipients that are in the formulation to remove API residue from the system. The advanced pharmaceutical manufacturing system is also fully automated, and it relies on continuous surveillance of in-process parameters, CPPs, through PAT and through the reliability of the instruments, as Owen showed. And we need that for real-time quality assurance. So PAT, this is one area where we have focused our energies in the idea of sampling. When you do PAT, all parts of your lot should have the same opportunity of being sampled, and the material should not be altered in any way after sampling. So to us, that is very important, and that is why we have worked a lot with real-time monitoring of continuous manufacturing. We started working at the chute after the continuous mixer, and we also did a lot of work in the feed frame. And monitoring in the feed frame is great because it provides you with an opportunity to use the same setup for monitoring in batch or continuous manufacturing. But we went ahead and developed a new interface for monitoring the powder blend, whether it is made in a batch or a continuous system. And that's what we call our stream sampler. The stream sampler is designed and patented by our group, and is designed to avoid particle rearrangement, attrition, and affecting the blend. Here you see it on the right side, the powder flowing through the sampler, and it is very smooth powder flow. It is designed so that all parts of the lot have the same opportunity of being selected as sample and avoid the complex flow dynamics of the feed frame, avoid powder recirculation. And the idea is that this could be used with all tablet presses. Okay, so the feed frame, this stream sampler can go after the blender. The powder comes in and goes counterclockwise 180 degrees later. The powder flows underneath a sapphire window where we can obtain an EIR spectrum or a Raman spectrum, and then it goes out at 270 degrees. So it is not recirculating the powder. Fresh powder is always coming through. There's a cup that can be used to remove material from the sampler to then bring it outside. Or the powder can flow to bring it to an external lab, or the powder can flow out at 270 degrees to a compressing machine. And this is something that we're focusing in, again, with the idea of providing a smooth representative sample where all parts of the lot has the same opportunity of being selected as a sample. This is now installed with our continuous manufacturing system in our laboratory, and we are now working, capturing all this data and putting it together. And we realized that the next step is to integrate it into some of the more common systems that are used to integrate the data from PAT sensors. So that's a lot of what we're doing. Again, that is the PAT part within this advanced manufacturing definition. We also see that advanced pharmaceutical manufacturing should be scalable. Okay, whether you want 100,000 tablets or 3 million tablets, it should be the same quality of product. And advanced pharmaceutical manufacturing should be portable. And the process should be implemented at different locations with similar results, regardless of where the product is manufactured. And portable also means in this case that process knowledge is transferred effectively from development to manufacturing. So again, this is our proposed definition of advanced pharmaceutical manufacturing. The PAT and the sampling related to the PAT is the work that we're currently doing in our group. And I think that's all from me today. Thank you very much. And my apologies for the accident we had here today.
Okay, great. Thank you very much, Rodolfo. And thank you for the great presentation and rejoining as well. And sorry for the technical issues that we faced earlier today. These things happen. It's okay. So we'll go back to questions then, and there have been some questions for you as well, Rodolfo. So we'll come to those as well. So we're just answering a question to Owen, which said, does Sentronic have any DMD-based PAT system? If so, for what purpose? Right as you rejoin. So we will move on to our next question for now, because we did answer this, didn't we, Owen? There was nothing we missed out. Good. Okay, then. So our next question, then, I'll ask it to Ahmed again. So this question, Ahmed says, just go down the list of questions. 1 second. So what is the opportunities and challenges of using spectroscopic PAT tools to monitor the granulation process and implement a control strategy?
Yeah, so I believe Owen is better to answer this question better than me. However, from my perspective, in the lab space, I would say interfacing PAT tools to the small, I would say, space of the granulation tunnel limits a lot of spectroscopic tools, and we have a very, very small window only at the end of the screw tunnel, but still we cannot monitor inside the granulation tunnel for different phases of the granules as it is formed. We just have used imaging at the end of the granulation tunnel, as I have shown, but space is always there, to explore and come out with better solutions. So, Owen, if you want to, add.
It's a really hard question to answer. There's a lot of things I could say, but a control strategy is a very, very complicated thing, and decisions you make about how you want to control a process early in the stage can have big impacts on how you implement PAT. The short answer is I would have a very long think and do my risk assessment analysis based upon what you have in the ICH guidances. And from there, based upon the output of that I think kind of puts you in a pretty obvious direction in terms of how you go for PAT.
Okay. Thanks, Owen and Ahmed. Before our next question, Rodolfo, did you have any additional comments to give to this answer?
No, that's fine. That's fine. Go ahead.
Okay, great. All right, then. Thank you. This next question is to Owen or Rodolfo, or both. So let's ask it to Rodolfo first. It says, how do you deal with materials sticking to the probe during continuous manufacturing? How do you ensure that doesn't happen?
How do I ensure that doesn't happen? I haven't had that problem because most of all that we've worked with are systems where the powder is flowing very, very fast. So I haven't had a situation like a wet granulation where something might stick. Everything I've worked with is very fast. If it occurs, you will notice because all of a sudden the results are very, very constant and very wonderful, and you have the most homogeneous mixture. That's all I can tell you. Sorry.
Okay. Thanks, Rodolfo. And Owen, anything you have to add?
Yeah, it's an interesting question. It depends on where you're making the measurement, and I think that the two responses I would give would be how you interface a probe to the process can have a big impact here. So if you look at some of the early papers on feed frame measurements from the University of Ghent, or talk to the people that do it from Thomas de Beer's group, the reason we go on the top of a feed frame rather than the bottom is if you put the probe on the bottom, you do tend to see more sticking. Going in the top, combined with the shear of material there, the likelihood of sticking to the probe surface, just looking at, a surface angle contact to sapphire versus steel, you are much more likely to stick to steel than you are sapphire. If you're sticking to steel in the feed frame of your tablet press, you're probably also picking and sticking and not doing a great job of making tablets. So that's one solution to it. There are other measurements, zones where that can be a problem, and I would say something like the GEA Lighthouse Probe's wash in place solution can be useful for at least slow buildup of material that could stick to a probe. Being able to wash that off with some liquid, then dry it, really dry it, is very helpful. So that's how I've seen this done in practice.
Okay, thank you, Owen and Rodolfo. Ahmed, is there anything you'd like to add here?
No, thank you. I think they covered it very well, yeah.
Okay, then, great. Thank you. This next question I'll ask to Ahmed. It doesn't say who it's for, so let's just see. It says, for continuous manufacturing, how do you perform batch yield reconciliation?
Okay. Yeah, so for continuous manufacturing, the definition of batch is not as a batch manufacturing. So it's mainly defined by the time running the system at steady state. Not by the amount of kilograms that's produced or throughput. So usually we use the throughput value for the batch at steady state, and monitoring the disturbances that may happening during a steady state by PET tools, and putting limits for any disturbance, how we can divert the system, and then coming again to steady state. All of this should be considered in batch reconciliation. But at the end of the day, throughput rate is the one that we are using for reconciliation. And as long as you can maintain a steady state, then you can have continuous running all the time. So there is some batches that's running for almost like a week or more, unstopping, as long as there is no disturbance that's happening there, and we have PET and control strategy in place to monitor it.
Okay, thank you, Ahmed. And any additional comments from Owen?
No, I think that's perfect.
Yeah, okay. And Rodolfo, anything you'd like to add here?
No, not right now. Thank you.
Okay, then. Good. Thank you, then. And this next question to Owen says, how does the spectral changes during the measurement of flowing powder, if the two different combination of integration time to coads used keeping the time for one spectrum same? For example, 10 ms and 100 coads, and second 20 ms and 50 coads.
Okay, sorry. I think I understand the question. So apologies if I'm off space here, but I'll answer to the best of my understanding. So the question is, what is the expected signal to noise change if you keep a constant measurement time, but you vary the integration time and then the coads to multiply together to get that constant time? The answer is going to depend on your spectrometer type, but let's assume you're using a diode array. It's what I've got the most familiarity with. That's going to depend on the fill level of signal on your diode array. This is very, very ballpark. We can talk finer numbers at a later time, do some math, but roughly above about 50% fill. So if the maximum signal in terms of, let's say, counts generated by your diodes in the center of the spectrum is 65,000 counts, if you're above roughly 32,000 counts, you stop seeing large improvements in signal to noise for the individual coads because you've significantly gone above the noise floor of the diode array itself. So once you're above that, then you want to start adding together coads. However, if in both situations you're above that, the number of coads, assuming white Gaussian noise, is going to scale, root n in terms of improvements to signal to noise. So the answer is it depends. 10 to 20, pretty big jump. If you're talking 10 versus 11, I wouldn't expect to see something huge, but there's a lot of assumptions in that. It would really depend on the particulars.
Mm-hmm.
Okay, thank you, Owen. And any additional comments from Ahmed?
No, thank you. I think that's fair.
Yeah. And Rodolfo, anything you'd like to add?
Fine.
Okay. Thank you then. Okay, so this next question to Rodolfo says here. Let's see again. Okay. Is Raman or NIR is Raman or NIR spectroscopy most common in PAT applications? What are the reasons for that? And what are the drawbacks of the lesser common technology, for example?
Okay. I think right now we have probably more NIR than Raman applications. A couple of things. For most of the time, NIR has been able to obtain spectra in less time. I think that difference between the two is reducing. It's being reduced more and more with changes in instrumentation. Usually, the noise level is less in NIR. But there are also applications where people want to follow crystal form changes. And in those cases, usually the Raman is better for those purposes. So obviously, it depends on your objectives and what you need it for. But I believe that both types of spectroscopies are very good assets nowadays for pharmaceutical scientists.
Mm-hmm.
Okay, thank you, Rodolfo. And Owen, is there anything you'd like to add?
Just a couple quick things. I think the folks at Lilly had some interesting papers here showing comparability. High level, my point of view tends to be threefold. Raman, the question's always going to become how much fluorescence do you have? How much could this change as a function of excipient lot? Measurement time, to Rodolfo's point, I do think that's harmonizing. And the third is just availability of process probe solutions. Folks have developed their own here, but if you're doing something commercially, it's often nice to have someone you can call to give you the solution. And I don't know how much process relevant stuff for something like a feed frame is really out there. So that's it.
Yep. Okay. Thank you, Owen. And lastly, Ahmed, any additional comments here?
Yeah, just one more thing is mainly about the sensitivity here for PET, especially for low, I would say, concentration materials. For example, if we want to monitor lubrication, for example, as it's affecting release in immediate release. So sometimes the concentration may not be picked up by near infrared, but Raman would be better. And sometimes the property of interest is more about a physical parameter rather than a chemical characteristics. In this case, Raman also may be better in this case. And so I would say it is mainly a function of the sensitivity or level of sensitivity of need, as well as property of interest that we need to monitor. But over the last 2 decades, both techniques and even other techniques like particle size distribution, Lezentic and FEBRM, for example, focus beam reflectance measurements, and BVM, for example, all of this is very useful in some application, and others maybe does not give better advantage. So I would say it mainly depending on sensitivity and property of interest.
Okay. Thank you, everyone, for those answers. So this next question to Ahmed says, what are the challenges observed in designing low API concentration, and what are the precautions taken to overcome those?
Yeah, this is a nice question. And I would say for the potent drug where the API concentration is very low in the formulation, so the blend uniformity and drug distribution would be the key here, and selection of the unit operation either in continuous or batch mode is the one to be selected in this case. For example, I may choose a certain milling process over the other. I would select, for example, certain drying process over other, fluidized dryer over, for example, granuloformer dryer or stereo dryer. So all of this is affecting, I would say, the distribution of API, and of course, having the measures to monitor blend uniformity and API distribution and content uniformity in different, I would say, unit operation and different intermediate product even would be the key to mitigate any, I would say, content uniformity issue during the line. And using, I would say, appropriate limits or acceptance criteria for each, either by PET or for continuous, for example, or even like chromatographic method for batch operation would be the key to mitigate any content uniformity issue in this case.
Okay, great. Thank you for that answer, Ahmed. Rodolfo, any additional comments here?
We have been able to monitor down to 0.5% weight by weight of an active ingredient, and some other groups have gone down to 0.1. It's always very important to do what we call composite sampling, which is to obtain a spectra of the powder as it flows and add a number of these scans. And you have to develop your method thinking of like, I need to take 16 spectra or 32 as the powder is flowing and average out over that certain area. And not have just one spectrum for each concentration, but also have multiple spectra for each concentration. And I think that is very important. And doing it again in a certain way where all parts of the blend have the same opportunity of being selected as a sample.
Okay, thank you, Rodolfo. And lastly, Owen, any additional comments to give to this answer?
Largely, I second Rodolfo's answer. I would add, I do think the perception of near-IR's limited detection, limited quantitation is probably a bit higher than it actually is. The Museo group, Rodolfo's group, other folks at Rutgers and other places have shown that it is probably 1% very doable. If you're smart about how you measure, you get representative sampling. I do think for something like a cancer drug where you're significantly below that threshold, there is an unmet need for PAT to work there. It's going to be difficult spectroscopically. If you're talking about 10 ppm drug load, that's a very real challenge from segregation. Getting representative sampling becomes very tricky. How you do CU, I think imaging might be an interesting option there. It's something like at-line, but that's a very much forward-looking solution, and I think it's going to be an active area of interest moving forward.
Mm-hmm. Great. Okay, thank you everyone for those answers. This next question for Owen says, can the Centro PAT be used in headspaces analysis to determine the concentration of oxygen in sealed pharmaceutical containers?
No, because we use a transflecting system, so that would be very hard to reflect off of there.
Great. Okay, very succinct answer. Thank you. Any additional comments here from Ahmed or Rodolfo?
Thank you.
Yeah. Okay. That's fine. I'll go to another question for Owen very quickly. It says, how far away can the fiber optic cable run on the Sentronic probes without losing significant signal intensity?
Good question. It depends on which probe we're talking about. The DRLS probes, which we are kind of latest and greatest, those are the feed frame probes we show off a lot. We support up to 50 meters. Anything above 30, I want to do an analysis on, but 50, we get the vast majority of the signal. The way we get that is by putting the light source in the probe itself. We do have some probes which do not have light sources in them. Anything above 5 meters on those starts to getting a little bit challenging.
Mm-hmm. Okay, thanks, Owen. Rodolfo, any additional comments here? Or Ahmed?
No.
Okay. Thank you.
Thank you, then. This next question for Rodolfo says, do you consider the use of a sample thief to be suitable to obtain a representative sample of a pharmaceutical blend?
No. No, because you're going after pre-selected sampling locations. And it's, again, for me, a representative sample. All parts of the lot should have the same opportunity of being selected as a sample. Unfortunately, it is still a regulatory accepted practice, and many processes are being accepted with sample thieves, but I do not consider it a representative sample.
Okay, thanks, Rodolfo. Any additional comments from Ahmed or Owen?
Yeah, traditionally the sample sieves are still used in batch operations, but of course it might not be suitable for continuous manufacturing. However, representation of the samples is very important, as Dr. Rodolfo mentioned. Like composite sampling is most important, even for PET. If we're not doing like composite sampling or spectral in this case, it may be misleading as well, right?
Okay. And Owen, anything more you'd like to add here?
Oh, there's lots I can say, but it would have a lot of 4-letter words in it. So I'm going to leave the other two comments.
Okay, then. Thanks a lot. This next question I'll ask this question to Ahmed. It says, what are the pain points in the industry right now in terms of PAT applications?
Oh, I think it's mainly the full control strategy, I would say, and using the PAT to control a process for feedback and feed forward. It may be easier for some, I would say, simple continuous lines, but when you come to a full integrated line like granulation, in addition to compaction and coating, for example, to have a full control strategy that can monitor the quality as well as divert, monitor disturbance, etc. I think we still like time to achieve this for complex processes. And yeah, there is a lot of efforts here in the agency and also in Rutgers and Puerto Rico and a lot of universities around working on this now.
Mm-hmm. Thanks, Ahmed. Rodolfo, any additional comments here?
That's fine.
Yeah, okay. And Owen, anything more to add here?
Shucks, sorry. I'm speaking to myself. Real quick, I would say training of personnel. There's an acute demand for people that have the skill set to do PAT and continuous manufacturing, particularly companies looking for people with a lot of experience there. And there isn't a whole heck of a lot of people that have 5-10 years experience doing continuous manufacturing because the technology is just new. So people that set up new lines want someone that has done it before and knows everything about it. And there just aren't a heck of a lot of those. That's changing. Folks like Rodolfo are putting out a lot of great people that are very, very knowledgeable of this. As the academy is picking up more and more of this, I do think that situation is going to get better. But from where I'm sitting, I'm seeing a lot of open hiring opportunities for someone with 10 years of experience doing this, and that's tough to find.
Mm-hmm. Well said. Okay, thanks, and for your answers there. This next question to Rodolfo says here, do you feel that the industry is moving towards the functional definition for advanced manufacturing?
Yes, I'm very impressed with a lot of the efforts in a number of companies. and all the ideas, and you have like nowadays the ICH guidelines, all the different groups that are trying to put together best practices. And yes, I think we're moving very nicely in that direction.
Thank you, Rodolfo. Ahmed, any additional comments here?
No, I am second to Rodolfo here, and even regulatory agency is encouraging advanced manufacturing and using of artificial intelligence and machine learning in all, I would say, operation. This is advances in the field, and FDA is partnering with many, I would say, industries and academia to increase our knowledge about this and to promote adoption.
Okay, thanks, Ahmed. And Owen, any additional comments here?
Fully agree with both other speakers.
Yeah. No, okay, then great. Thank you. So just a couple of questions left. We can get through them before we finish. This next question to Owen says, how does the single scan and the coads work, as the lights will be continued during multiple coads? So what will happen during single scan and coad in terms of instrument/detector function?
Yeah, good question. So the light is always on, so we're not flashing the light. So that's just a continuous source. In the case of a single scan, or single measurement, we're not scanning anything, keep forgetting that. We would collect a single accumulation on your detector and then report that out. That report time is non-trivial, so on the order of 10, 20 milliseconds. In contrast, when we do coads, we would continue to coad and actually store that in a buffer on the hardware and then report that out. That buffering time doesn't add anything to the measurement time, but there is a flat write-out time of around 20 milliseconds. So hope that answers the question.
Great. Thank you, Owen. Rodolfo, any additional comments here?
Well, that I think is more operational to the instruments so that he has. So I cannot say anything else.
Sure. Okay.
I'll leave it to Owen.
Yeah. No, I don't know. Ahmed, anything you'd like to add here?
No, I cannot agree more with Owen. Yeah.
Yeah. Okay, great. Well, our last question for today goes to Owen again. And this question says, is there any impact of exposure time/integration time of near IR on depth of penetration in powder sample?
Oh, boy. I have not seen evidence to indicate... Well, so I'll start with the easy one. Exposure time, integration time, I would not expect anything to be there. There's an adjacent question around illumination intensity that is probably a bit more controversial. I have heard people say that increasing illumination intensity will increase depth of penetration. I am skeptical of that. I have not seen data to support that. If I do see data, I'm open to changing my mind, but from a physics perspective, for a loosely packed powder or diffuse reflection measurement, I am skeptical you're getting significant improvements in depth of penetration as a function of lumens put on the sample.
Yeah, the problem also is if you get higher depth of penetration, what you may obtain is transmission of the radiation. It's going in the opposite direction where the detector is not going. So after a certain depth of penetration, the radiation just isn't coming back. So it's a very interesting physics phenomenon. I think we've been studying scattering for over 600 years. So if you want a vocation in life and you still haven't found it, light scattering is my recommendation. There's a career for always there.
Okay.
Yeah, it goes back to the traditional, always work.
Yes. Yes.
Okay, great. Is anything else to add, Ahmed? Or happy for
Oh, thank you. I agree with Rodolfo knowing so much.
Yeah, good. Well, one more question just popped up, so we'll try to get through this very quickly. I'll ask this question to Rodolfo. It says, how can we evaluate the correct implementation of PAT in continuous manufacturing?
Wow. Oh, wonderful question. I think it's just, all the... I'm really impressed. I've been meeting with people from industry, and I'm very impressed about the systems that they have implemented to, in terms like their IQ, their performance qualifications, their operational qualifications, all the steps that they do to make sure that a system is working well. The ideas of a system suitability test, as Owen suggested today also. I think it's just, you have to look at the data carefully, and you have to prepare yourself to work very carefully. And I think people are doing a lot of that nowadays.
Mm-hmm. Great, thank you, Rodolfo. Ahmed, any additional comments here?
No, I think we're just trying to bring the PAT very close to the validation of chromatographic validation, which is very, very sensitive material. So we are trying just to follow the same, I would say, validation steps that's used for chromatography by applying PAT in this case. But at the end of the day, it is just ensuring that what we are looking for, we are getting it.
Mm-hmm. Great. And Owen, anything more to add here?
No, I think those are great answers.
Yeah. Great. Well, that was our last question for today. So thank you everybody who sent in their questions. It's quite a busy Q&A session. I always enjoy those. Thank you. And please accept my apologies earlier for the technical difficulties that we had with Rodolfo as well. But all of this will be available to replay again later as well. So you can watch this webinar and other webinars by going to our website, which is biopharmawebinars.com/webinars. And before I finish today, I'd just like to go around our panel of speakers here and ask if there's any concluding takeaway remarks. So Ahmed, over to you first.
Thank you. First of all, I want to thank organization and coming together with Dr. Rodolfo and Owen for this webinar. And the concluding remarks that better understanding of the process and controls is the way to come out with I would say successful product and process.
Great, thanks, Ahmed. And Owen, over to you next.
I'll second Ahmed's thanks for the opportunity. I want to thank Ahmed and Rodolfo for their wonderful talks. I learned a lot. That was really insightful. Closing remarks on mine. High level, I think the PAT community does a wonderful job of proving PAT works for OSD manufacturing. I think now it's time to buckle down and do the yeoman's work of doing it reliably, making sure it's really ready for a real-world manufacturing environment. I think folks like us have a lot of work to do there, but I would advise people considering continuous manufacturing just to keep that in mind, even if it's not the most exciting thing to talk about. It is something that is important.
Thanks, Owen. And Rodolfo, over to you.
I'm very happy to see the progress of PAT and continuous manufacturing, and hopefully we share that progress today. Thank you.
Great, well said. And I would also like to thank our presenters for joining me here today. So once again, that was Dr. Ahmed Zidan, Senior Staff Fellow for Center for Drug Evaluation and Research at FDA. So thank you, Ahmed. Also, Owen Rehrauer, who is President at Sentronic US Corp. So thank you, Owen. And last but not least, Rodolfo Romanach, PhD, Center for Structured Organic Particulate Systems at the University of Puerto Rico. So thank you, everyone, for joining me today. It was a great webinar.
Thank you. Bye-bye.
Bye-bye. Have a great day.